Look, I got a bit confused. There were quite a few questions asked, and so I'm just going to quickly go through a few that are prepared, but they did include the four that Alison just quickly show across. So one of the questions was, could we explore alternatives such as gene editing by incorporating genes from other plants with enhanced defense mechanism instead of relying solely on pesticides or prey predator methods? Look, there are challenges for gene editing palm crops. So there is a very nice short review paper or opinion paper that came out in Frontiers in Plant Sciences, and that just came out literally last month, a month ago. So it's here. I encourage everyone to have a look, have a read, if you're so interested. Specifically for Figure 1, it tells you the various challenges to use gene editing to improve and protect the palm crops. The palms considered were the betel nuts, the coconut, palm oil, and date palms, amongst others. And essentially, the genome of the plants, of the coconut plants is quite challenging for assembly. So you need the genomic resources to enable you to do some editing. But also specifically the long developmental stages for the young plant to grow up, as well as identifying the genes of interests of the specific trait that you want to promote and to culture. So these are various challenges. So it is currently being explored to a certain extent, but nevertheless, it's still a fairly new area. So for example, in the section here, I just highlighted a bit. So there's a few genes here that are already being identified, but they're also suggested that it could potentially be targeting the insects as well. So these are quite interesting, and specifically for coconut rhinoceros beetles, various genes have been identified. At least 5 different genes have been identified to cause mortality at the early RNAi injection stage. Ultimately, there is a question of the social and cultural aspects where GM crops, how widely and how willingly would the community accept it, and also the trading of GM commodities as well. So these are all considerations that needs to be explored. There was another question about specific pheromone lures. I know that Mark had looked at the trying to identify CRBG specific pheromones, and they were using the Solomon Islands population. And as I presented my talk last time, we found that there is no CRBG, the clade 1 specifically on Solomon Islands, and that may explain why they failed to find any specific pheromone, and the identification of the CRBG could be due to the fact that they use a very short DNA sequence, and that was a suboptimal molecular markers for identifying the Guam specific population. There are, however, a lot of pheromone products out there, so you just have to do a Google search and you come across both video and you can also find the products itself. In terms of elaborate on nudibranch control of CRB, I don't have any specific experience on that, so I'm just going to say that I just have to I read the literature, and there's quite a few nice one, and Jeff Bedford, a retired researcher in Australia, had done a lot in the Pacific, and his papers are good to read. So I encourage everyone to revisit the historical literature. Have you tested the other biomarker genes other than CO1, for example, ND1? So yes, we do have a paper, as you can see, that is literally being submitted. This paper had been prepared for a while. We've shared some of the primers, alternative primers with some of our collaborators, for example, from New Zealand and Philippines. And essentially, we developed the primers on ATP6 and CO3. So if you're interested, please get in touch. I'm more than happy to share the primer sequences with you. And you can see that based on the CO1, you just get a general clustering of the various populations. But however, if you use the gene and the gene region that we identify, it literally separated out uniquely Guam population from the others, which are also quite geographically distinct. So these are good, we think, robust primers for testing it. Now, question 5 and 6 relates to the CO1 gene between samples. So, so the question is, what are the fundamental, would there be any fundamental reason for variation in CO1 genes between samples if they do not denote completely different haplotypes? And also the other one that are related is on your slide showing variation between samples when analyzing a bigger section of the full genome. Does that mean that we're dealing with several haplotypes than just CIBG and CLBS? Look, this question is basically come down to how we define CO1 and the haplotypes. The definition in general, haplotype is just a genetic variant. And it literally represents a variation between individuals. So you can have, for example, not only if you're just looking at CO1. So let me go to my, this slide, which is what was referring to. So if you recall, this is considered as a single haplotype, mitochondrial DNA haplotype for the CO1, okay? Because they are all identical sequences. So this would be one haplotype, all this would be one haplotype, and this would be one haplotype. So in here, we have 3 CO1 haplotypes. However, when we look at across the full genome, we could easily have picked this region, and in which case, there are 2 haplotypes. So haplotypes are just variation. So how many are we actually dealing with in the population? I think this is a good question, and I don't think we can ever finish serving the population diversity of the haplotypes. So just let me go back to the question now. So does that mean that we're dealing with several haplotypes? We're dealing with many, many haplotypes. And in terms of the CIBG or S, the definition, you just have to remember that these are based on mitochondrial DNA. And there is actually no evidence to demonstrate that whatever mitochondrial DNA haplotype that has been identified, there was a strong association with resistances. So essentially, you can just forget about the G and the S, in my opinion, based on mitochondrial DNA, because if it were to be a resistance, it's likely to be both a contribution from a father and a mother. And therefore, you really need to look at the nuclear DNA. So having a mitochondrial DNA where it doesn't reflect the paternal side is a bit meaningless. Okay, so we are dealing with many, many different mitochondrial DNA haplotypes, but these are just population diversity, and that's what there is to it.
60 seconds, Jake.
Okay, and finally, the climate change. I think you need to really swap this. There are a lot of literature that talk about climate change and genomics. In terms of coconut variety play a role in genomic evolution, should be the other way around. Genomic evolution leading to coconut varieties. That's it.